This photograph marks the announcement of the Human Genome Project, the first time we successfully read the billions of chemical letters, the A's, T's, G's, and C's that compose human DNA. But mapping those letters immediately created a massive bottleneck. Sequencing generated a dataset so vast that it is impossible for a human to read or process without assistance. The complexity of the raw output is evident in this chromatogram. Simply reading these chemical peaks doesn't explain what the genes are or how they are physically organized. This problem led to the growth of bioinformatics, a field staffed by scientists trained in both biology and computer science. These researchers developed the software and algorithms needed to store, manipulate, and decipher these massive biological datasets. Today, analyzing life at the scale of billions of base pairs requires these computational engines just as much as it requires the physical lab. This technology is how we know that humans share 80% of our protein-coding genes with a laboratory mouse. An algorithm scans the human genome for a specific string of letters, like ATTGCACGTCT, looking for an exact match in the mouse genome. Once found, the algorithm scans past the sequence ends, pinpointing where the two genetic codes diverge. These points of difference help researchers identify the genes responsible for unique human traits, like our specific brain development, or the fact that we lack a tail. When sequences match perfectly across different species, they are known as conserved genes. Humans share these conserved genes not just with mammals, but with insects like the fruit fly. We even share them with microscopic bacteria. Because these genes are shared across so many species, they likely code for proteins that are essential for all life on Earth. By computationally scanning for these matches and deviations, bioinformaticians can isolate the exact code changes that evolution used to develop different life forms. These same tools allow us to compare the differences between individual humans, which is a critical step in understanding disease. Researchers use genome-wide association studies to scan hundreds of thousands of genetic sites across a population. Suppose we look at 50 healthy people and 50 people with diabetes, searching for a single base DNA difference. If 47 out of 50 diabetic patients carry a specific A variant, while only 5 healthy people do, the software flags a strong correlation. This data indicates a strong association, though it does not prove that the genetic variant is the direct cause of the disease. These computational findings serve as a filter, narrowing the search from billions of bases to the specific regions where biologists should focus their next round of lab experiments. This analysis is especially important in oncology. Cancer is driven by mutations and changes in the nucleotide code, causing cells to multiply chaotically. As sequencing costs decrease, doctors can run a direct comparison between a patient's normal genome and the genome of their tumor cells. Algorithms cross-reference these mutations against massive databases containing the tumor profiles of thousands of other patients. This identifies the precise DNA changes that are causing that specific tumor to grow. This analysis provides a molecular target, allowing doctors to select therapies that match the specific genetic mutations present in a patient's tumor. As genome sequencing technology continues to accelerate, the size of these biological datasets will grow exponentially. This massive growth requires bioinformaticians to develop increasingly high-speed algorithms just to handle the influx of new information. The challenge for the future is to expand these centralized databases to drive medical discovery while strictly preserving the privacy of the patients involved. Future medical advancements depend as much on the security and speed of these algorithms as they do on the biological discoveries themselves.